Good afternoon to all of you. My name is Elsken van der Wal and I'm the current chair of the Strategic Program Cancer of the UMC Utrecht. And it's a great pleasure, but most of all a great honor, to welcome you at the Associate Professor Lecture of Dr. Maria Rodriguez Colman. And before I give the floor to her to give her lecture, I would like to give a brief introduction about who is talking this afternoon. Dr. Rodriguez Colman was born and raised in Argentina. She obtained her PhD, granted with honors, we would call that in Dutch cum laude, at the Institute for Biomedical Research at Lleida University in Spain. The subject of her PhD research focused on how cell cycle transcription factors regulate nutrient metabolism and redox stress in yeast. Following her PhD, she continued working as a postdoc in the same institute, but happily for us, she then came to Utrecht, and she took up a position as a postdoc at the Center of Molecular Medicine. Here at the UMC Utrecht, also called CMM, in the research group of Professor Boudewijn Burgering. At CMM, the focus of her research has become on how metabolism regulates stem cell dynamics in healthy tissue and in cancer. As a PI, she now has built her own group within CMM, called Metabolism, Stem Cells and Cancer. And this is a very international composite group, with researchers from Vietnam, Egypt, Croatia, Germany, and a little bit of the Dutch. I'm sorry. She's one of the pioneers in implementing organoid technology to metabolics in stem cell biology. And her group has adapted and developed techniques such as bioenergetics analysis and 4D live imaging of metabolites and cell signaling in organoids. Dr. Rodriguez Colman is author on several high-end papers, and she's an invited speaker at both the national and international podia. And to promote the science at another level, she is also involved in the network of Argentinian scientists in the Netherlands, an initiative from the Embassy of Argentina. She has been honored with very prestigious awards, amongst which is, and I do hope I pronounce it correctly, a mention d'honneur in 2018, granted by the Honorable Senate of the nation of Argentina. This is an exceptional award, which is granted to professionals that have excelled in specific areas, such as science. In 2021, she was granted an Aspasia Award by the Dutch Research Council, also called NWO, in recognition of the excellence of her scientific work. In that same year, she was appointed Associate Professor at Utrecht University. And today, this appointment will be honored and celebrated by her lecture, which we all look very much forward to. And again, I cannot say it enough, it's a great pleasure, but most of all a great honor, to give the floor to Dr. Rodriguez Colman. Maria, the floor is totally yours.
Thank you, Eske, for your very kind introduction, and welcome you all to this online lecture where I'm going to share with you our research at the UMC Utrecht. Today we're going to talk about metabolism. And what is metabolism? Metabolism is the compilation of all those processes that ultimately support life. Here I would like to share with you this beautiful picture with this nice landscape with these two siblings. And this transmits peaceful, it looks very quiet. However, from the point of view of biology, a lot is going on. The simple fact that the boy is sitting straight or the girl is leaning into the water, that is supported by the organization of several different cells in our bodies that actually enable movement through muscle contraction. So how is that possible? It's because of metabolism. Metabolism is all those reactions in which we have a transference of energy or mass. If we take us humans as an example, metabolism is involved with our uptake of food and water. So how do we convert food and water into energy? We have these very specialized functional units, which are our organs, and each of these organs have specified to comply a specific function. So when food gets into our bodies, digestion will start, and it will be developed mainly in the stomach, and then at the point of the intestine, we will have the uptake of the nutrients. Then the liver will work as a sort of storage, but also will regulate metabolism systemically in the whole body. We also have lungs, which are very fundamental because that bring oxygen to our bodies and enable several of these metabolic reactions. And we have elimination of waste, very important, which we rely on our kidneys for it. Interestingly, in each of these organs, there are several cells that make them. And we have not one population of cells, but different cell types. We can classify those cells in two main groups, stem cells and specialized cells. Stem cells are those cells that actually can divide and give rise to more cells, and those are the reservoir for our tissue regeneration. At the same time, these stem cells can become specialized in different functions, and all together will enable our organs to perform complex functions. Interestingly, at the level of each of these cells, there is also metabolism and metabolic reactions. This looks very confusing and complex, and that's actually the point of it. This is a scheme of all the metabolic reactions that take place in every one of our cells. You can see here, each of these dots is a metabolite, and the line that connects two dots is a metabolic reaction. We can see also in different colors, different metabolic pathways. So you can see how everything is interconnected and the high complexity of these reactions. And all this is required to perform very specific functions. Today we're going to focus on energy metabolism, which involves those pathways that are important to make energy and support our life. If we talk about energy metabolism, we have to talk about glucose. Glucose is a very simple molecule. It's the most simple of the monosaccharides, of the sugars. And it's very interesting because glucose and the way we consume glucose to make energy is highly conserved. And that is that at all different levels of life, yeast, bacteria, plants, animals, and here a nice human having glucose, we all consume glucose in a very similar way. In our body, in humans, regulation of glucose is strictly controlled. You may be familiar with this, as this regulation of glucose in our bodies or of insulin will lead to disease like diabetes and consequently also problems in the heart, coronary problems. At the cellular level, glucose is consumed in two main different ways. One is glycolysis, but basically that provides energy and also it provides mass, for instance, to replicate the DNA and make new cells. The other pathway that also produces energy and uses glucose involves mitochondria and also provides important intermediates for the growth and replication of cells. So all these two pathways are not mutually exclusive. And if we think about differences between different cells, we also have to acknowledge that different cells can have different metabolic phenotypes. And that is quite well understood, that different cell types, different types of cells will have different metabolic phenotypes. However, could this be the other way around? Could metabolic changes have an effect in the fate of the cells to determine what a cell will become, for instance? So that is part of what our research is really interested in. We want to understand how specific metabolic changes could have affected the decision-making of the cells. Those could be, for instance, if a cell is going to become specialized, or it will remain as a stem cell. But it can also have a very big implication if a cell will, for instance, die or transform. And transform means go from a healthy state to a disease state. And in our lab, when we think about disease, we actually mainly work on cancer. And it all begins in the intestine. Why? Because the intestine is the first surface, as the main surface where the nutrients are taken up from the food into our bodies, inside our bodies. That also means that our intestinal cells are constantly facing changes in metabolites because we eat or do not eat, or we change what we eat. So we do know the intestine enables consumption of food and consumption of nutrients and metabolism, but can the composition of what we eat affect the fate of intestinal cells. And that actually has been already researched because it's such an interesting question, of course. One of the studies I want to share with you is the studies that relate to calorie restriction. The idea of this research was to evaluate whether we eat less, if that would have an effect in our intestine. So this type of research is normally done with mouse models. And here you can two types of mice. One group of mice was fed ad libitum. That means that they will eat as much as they want. And the other group of mice will be restricted in how much they can eat. These mice, the people looked at the composition of the intestine, and they looked how the intestine looked like, and they actually found the amount of stem cells, the amount of the reservoir of the cells that keep the regeneration in the intestine has increased. And a later study actually related that to cancer development, under the assumption or the idea that when you have more of these healthy, normal stem cells in the intestine, that could prevent that a cancer cell could implant there and generate a tumor. So in that respect, this type of studies will suggest a positive effect by decreasing the chances to develop cancer. Another interesting example of this type of research is about high-fat diet, which is also normally called as the Western-style diet. In this case, mice was again fed ad libitum, as much as they want to eat of a normal diet, and compared to mice that had fed with a lot of fat. Here, again, when looking at the intestine, they also find an increase in the population of stem cells. But in this case, what they found is that because the abundance of fat was so high, the cells went from consuming glucose to consume fat. And that actually seemed to have a detrimental effect by increasing the chances of developing cancer. In general, what is clear so far is an ongoing and very hot topic of research is that diet does have an impact in the intestine and the chances to develop cancer. So either by caloric restriction or moving glucose to fat, increases the proliferation of the cells. Our research in our group, we have actually shown that not only a change in the type of nutrient, but also how this is consumed could also have an effect on the cells. Actually, we found that cells will become specialized if they move from consuming glucose from the glycolysis to mitochondria. So, and these two facts, proliferation and specialization, are the two key factors that actually altered and are the fundaments of cancer development. Why? Because normal cells, healthy cells, will have a very strict regulation of when to proliferate or not to proliferate, or when to become specific. However, in cancer, this is completely disbalanced. So cells lose their sense and they start to proliferate out of control. And in that way, they promote tumor growth. And actually, this happens because of mutations in the DNA that actually will drive this dysfunctional behavior. What does metabolism have to do with this process? Well, actually, if we have it following what I have been saying, metabolism will enable and support energy and giving mass to allow that these cancer cells will grow out of control. And I would like to share with you an example of this. We can think of a tumor growing out of control as a train which is riding out of control. If we think, why is it riding out of control? Because something is broken in the system. Which is the defect in the engine will be, for instance, comparable to the mutations in the DNA that promote that behavior. Now, if we actually think, how is that enabled? How is that supported? Because of the fuel. Fuel is at the ultimate level why the train will not stop, and that is metabolism in the tumors. So following that rationale, we can think, okay, let's stop the fuel and we kill the tumor. So can we target cancer metabolism as a strategy to fight cancer? And actually, this is already ongoing. And this is based on one of the examples I want to present is based on a specific type of molecules, which are the antimetabolites. Antimetabolites are actually molecules that really look like a metabolite, very similar. However, once in the cells, they do not allow a metabolic reaction. So they take the place of the normal metabolite, but do not allow the metabolic reaction. And those are, for instance, examples of capacetabine, 5-FU, or methotrexate. Those antimetabolites interfere with the synthesis of new DNA and do not allow the cells to divide. But there are certain limitations, and that is, among others, is toxicity. And why is toxicity? Because these reactions that these antimetabolites block are not only required for the cancer cells, but are also required for the healthy cells. And that is why we cannot go too high with a dose, for instance. So we should think of ways of metabolic characteristics of a tumor that are not shared with the normal cells. And there are a few. And we can come back to glucose, because cancer cells, differently from normal cells, are glucose addicted. And actually, they consume much more glucose of normal cells, and they consume it in a way of glycolysis, and they produce high levels of lactate. This phenotype is so highly conserved that actually serves as a prognostic or a diagnostic in the clinic. You may be familiar with PET scans. Using FDG, which is an analogous of glucose, is actually lighting up the tumors based on the high uptake of glucose. So another interesting aspect of this cancer metabolic phenotype is that this phenotype is highly conserved and highly studied. It was first discovered about 100 years ago. However, we still debate why would these cells engage such a metabolic transformation. So why? And there are several interesting aspects of why cancer cells will behave this way. One of the ideas is that to survive hypoxia, it is believed that if cells cannot reach oxygen levels for their functions, if they can rely on this pathway and not using mitochondria, that could be sufficient for them to survive. And most recent idea, which is also very attractive, is the idea that these high levels of lactate, which are produced for the high uptake of glucose, will make an effect on preventing the immune system to attack the tumor. And that is either because immune cells and tumor cells compete for glucose in the environment, or because the high lactate concentration does not allow the cells to mature and efficacy target the tumor. So if we go now to our own research, what do we do with cancer metabolism? How do we study this? Well, we want to understand the fundament. We want to understand the house. For that, we need a very controlled system. So we use organoids. Organoids are relatively derived from adult stem cells, are relatively newly established, actually from Utrecht, the first work on this. And organoids are obtained from biopsies from colorectal cancer patients in this case. And this can be grown now in vitro in very controlled situations, and they will quite faithfully recapitulate the development of a tumor in the patient. Because we're interested in monitoring metabolism in the cells, but also if they will change their decisions of specification, of dying, or of becoming a stem cell, we also introduce some tools genetically to be able to visualize metabolism and also to visualize cell identity. And we use microscopy. Here is an example where we have the different colors to monitor the two different parameters. This is just an example to share with you the type of data we look at. We can see here in the first panel, we can see the growth of a tumor organoid, and we can monitor metabolism, or we can look at cell identity, or we can look at both combined. You can see that we can monitor the development and all these changes on time. We can extract this data. We do this in collaboration with the biophysics department in AMOF in Amsterdam, and we can extract this data, and we can not only then visualize, but also quantify different parameters of tumor biology. We can calculate growth rates, but also we can follow metabolic changes, or also if cells lose viability or they die. What is what we want to do with this? What we actually do, we like to mimic different metabolic environments, having in mind diet. So we want to see whether changes in the glucose in the diet, or proteins, or fat, or even mimicking intermittent fasting could have an effect on how the tumor develops. We're also quite interested to see how these tumors react to chemotherapies. So we can combine the conventional chemotherapies, that's something we are actually doing, with metabolic targeting drugs. And in that way, we can see whether we can improve efficacy of these drugs. And of course, we combine this type of technologies with other ones like based on biochemistry, molecular biology, and several omics, because at the end we want to understand how these processes are regulated. As you may have appreciated, our research is basic research. That means that we want to understand how nature works. We want to understand disease. We are at the very basis of levels of research. We can see in this scheme the different basic steps of research. We have basic research. We have translational research, where we can address whether our findings can be implemented to treat disease in models. We can also move that into clinical research, where we can address whether these strategies could be valid for human treatment. And at the end, of course, there is implementation, where the whole society could benefit from science. We are the very first step here, and that's why I consider basic research also is called fundamental research. All these different layers are important. And actually, this arrow is not a unidirectional, but should be a bidirectional arrow. But actually, the best of research comes from interdisciplinary research, but also with a high crosstalk between the different layers of research. And in that respect, the UMC is very strong, because we actually can cover all these different levels of research. So I expect in the coming years that our research, very fundamental research on cancer metabolism and metabolism of stem cells, can provide some benefit to society by hardly collaborating with all these different layers of research. I would like to thank our funding and supporters, and of course, a great group of people that we work with, because research is a team game. It's not one person working, but it's really about the team. And I'm very lucky to have a nice group of very talented and enthusiastic researchers that I lead and supervise in that every day. Thank you.
Well, thank you, Dr. Ulrike Colman. This was a very elegant and beautiful talk. I wish I had you as a teacher when I was a student, because now for the first time I understand metabolism, the role of glucose, and I know that I need to be careful with eating fat nutrients, but that's not a thing. And I love your last part. I think that's very important, that we do this interdisciplinary, multidisciplinary work. And I think the way you showed us, like, this is the research that I do, but you make the connection with the chemotherapy that we administer very frequently. You showed the PET scan. I mean, this is lovely. And I really am very impressed. Thank you very much. And I wish you all the best with your group and your own research. Thank you.